I want to introduce our expert on weevils, and his name is Jeremiah Vardiman, right?
Correct.
Okay, you're on.
All right, thank you. Yeah, Jeremiah Vardiman with UW Extension. I'm located up in the Powell office, but I cover the state as need be kind of thing. Chance Marshall, you guys know him, he invited me down, wanted to highlight what we've been doing with some alfalfa weevil research, so I wanted to give you guys the kind of the update or what we're seeing out there. If you're in here to get the answers of what to do, I don't have those answers, unfortunately, and I don't think the scientific community has that. But we'll give you what we see. We're glad to talk through some things for you, talk through some strategies and that. But anyway, let's just get into it. For most people, I'm going to grab the lights here. Might be able to see the slides a little better. Most people probably already know this life cycle, but just wanted to touch base, make sure we understand what we're talking about here in Wyoming. It's a little bit different depending on if you talk in different areas of the state. So for us, for strains of alfalfa weevil, we only have one. We know we have the Western alfalfa strain. So strains are like a variety of weevil, right? Or species of weevil. They're called strains in the insect world if you're an entomologist. We have the Western strain. Okay, so there's also the Eastern strain, which is usually found around Oklahoma, Kansas, Texas, kind of back there in the east. There's also the Egyptian strain. That's found more in Arizona, New Mexico, Southern California. And the big thing about those different strains are, here in the Western, with the Western strain, we only see one life cycle typically within a season. Okay? Those other strains, they're going to see multiple, usually two is most common. If you get up around southeast Wyoming, so you're going to get up there in the Panhandle of Nebraska, eastern Colorado, they kind of have an overlap of that eastern strain and western strain. And when you look at the map, everybody will show you, oh, well, this is where those strains are at. It's really not very scientific. It's like somebody took a crayon and just kind of drew some areas of where they're at. So just wanted to make that clear. But you might hear of like the Egyptian strain like that. And that's why I want to highlight that, is I've even heard up in the Bighorn Basin, a producer told me that they were wondering if they had resistance in their field. This was 2 years ago. The agronomist that was there for the local cooperative, or not cooperative, but the local spray company, told them that we have a new strain, the Egyptian strain. That's why you got to go to a different chemical. If you hear that, that's not true. We have one strain that we are aware of, the Western strain. And it doesn't matter what strain you have, those chemicals are still effective as long as we haven't built a resistance to them. Okay? But we'll go over this life cycle. We're here at this time period. You know, this is kind of viewed from September to March, and that's the overwintering sites. Where is that found? That's in the field. Base of the crowns of those plants that are left in the field, if you have any stubble, they might move to the edge of the field. There's some literature out of South Dakota that really highlights that those adult alfalfa weevil are moving out of the field to the edges of the field. There's research out of Utah suggesting that alfalfa weevil are mobile. They actually have functional wings under the elytra, and they can travel miles. And they're moving up dry canyons. So if you are bordering BLM, rangelands, that kind of thing, we don't know, but it's possible our weevil are moving out of those fields up into the badlands or up into the range country to overwinter. So they're not always in the field. Those overwintering sites are different, and kind of to about March. Once our springtime temperatures, and it might be this month, who knows, February this year, the way it's going. Once we start hitting daytime highs of 48 degrees Fahrenheit, kind of consistently, if you're seeing 3 to 7 days, that's probably enough warm up for those adult weevil to start getting active again and start moving around. Typically, we see that about the end of March, beginning of April. Like I said, this year, it's anybody's guess. We might see a little bit more activity sooner in the year. What that means is those adults are going to start migrating. If they're not already in the field, they're going to start migrating back towards their fields. Okay, that's when breeding's going to happen. Our adult weevils, are going to breed in the field and start laying eggs. They're going to chew a hole either in straw that's left over from last year or new growth in that alfalfa plant. They're going to chew a small hole in there, and the female's going to deposit eggs in there. Typically within one laying, she can lay like 20 to 30 eggs at a time. In her lifetime, or that spring, she will lay up to 500 or 800 eggs in a field. So it doesn't take many females in a field to get us to high population levels that'll impact our alfalfa, right? So that's what they're doing about April. Into May, we're starting to have egg hatch. We're having larval development coming out of those eggs. They're going to come out of the stem, and then they're going to crawl to the tip of the alfalfa plant. And so they're going to bury themselves down into the whorl of the alfalfa plant leaves at the very tip, that terminal bud that's growing. I should mention, when they're in the egg form in the stem, when they're in the whorl of those leaves, they're protected. So if you did a chemical application, you are not going to be controlling anything. Okay, you're going to be wasting your time, energy, and money. As they're living with inside those tips of those leaves, they're chewing on the new leaves that are being developed on that alfalfa plant. Okay, these are considered first instar stages of the larva. They're a little grub. They're a green. Can't really see them very good, but there's two of them right there. But they're very small green larvae that have a black shiny head. No other larva has that characteristic. So if you're seeing green, a lot of times producers will tell me they're worms. They call them worms in their field. They're a green worm with a black shiny head. That's an alfalfa weevil larva. Okay? As they grow and develop, they're going to go through 4 different instar stages of growth and development within the field. The first 2 are in the whorl of the leaves. And the first instar, they're smaller than the head of a pin. So like a push pin, that head, they're smaller than that. They're very, very small. They're hard to see. You can see them with the naked eye, but they're very hard to see. Also, when they're in those whorl of those leaves, if you're out there doing any sampling, if you're doing sweep nets or doing a bucket to try and look for them, they're very hard to dislodge out of those tips. You almost got to tear those tips apart to find them. So a lot of times we don't see the first instar stages. They eat enough, they grow enough because of temperature, because activity, they're going to grow and grow up into a second instar. Scientifically, this is way more than you guys probably want to know. This is what entomologists do in laboratories. They actually determine what instar stage those alfalfa weevil are in. They measure the head capsule of the larva. And I can't even tell you what that differentiation is. I can look it up. But all I'm saying is a lot of times people look at the size of the larva to determine what size they are or what stage they're at. And that's a rough indication, but to the literature it doesn't correlate. Okay? It's the size of the head capsule, not of the entire larva itself. By the time we hit into second, beginning a third instar stage, that alfalfa plant has grown up enough and those leaves are starting to uncurl, unfold, and push out. And that's generally when we start seeing them. That's when producers start noticing them in the field, or crop advisors see them in the field. Third and fourth instar stages is when the most damage occurs. So they are physically defoliating the plant. They're skeletonizing the leaves is what it's called. They do not eat the veins of the leaves. That's too tough of a tissue. They're eating the alfalfa tissue between veins. And so then all of a sudden you have what's left is the skeleton of the leaf. The veins of the leaf are what's left. If it gets severe enough. Okay? So that's happening about mid to end May into June. This is all coinciding, so third and fourth instar stages, the peak populations are all coinciding typically with our first harvest. And it's usually only a problem in first harvest. If we can get through first harvest, we've usually dodged the bullet. I'll give you a couple examples where that doesn't always occur, but that's our first life cycle. Once they hit fourth instar stage, they then migrate to the base of the plant. Down again at the crown, at the ground, they spin a very loose cocoon and pupate. They go through complete metamorphosis and turn into that snout beetle again as an adult. When they're in that cocoon, again, they are not vulnerable to pesticides. Okay, that those cocoon has a water barrier or waterproofing, and so any chemical that's applied just kind of rolls off that cocoon and it protects that pupil inside. After that, so we're at kind of the middle to end of June, beginning of July, we're having adult weevils coming out. The adults that were in the field laying eggs are starting to die at that point. They've reached the end of their life, and now we have new adults in the field. They go into summer dormancy for July and August, mainly because of the heat. Once we start kind of cresting over that 90, 95 degree temperatures and up, they just don't do much. They don't like that heat, and they just kind of go into a slow dormancy stage. They hang out in the field. They might move to the edge of the field again. They're not eating much alfalfa. They do eat some, but it's not to the levels that we see any damage. And so the adults are not really considered a problem or a pest in the field, other than they lay eggs for more larvae for the next year, right? And then once we hit about September, those temperatures start cooling down again. Those adults are going to then start finding their overwintering sites, and the whole cycle starts over. Now, I'm pretty sure you guys are just like we were up in the Bighorn Basin. It's been a long, beautiful fall, mild temperature, right? In these type of years, those adults can actually get active and start breeding. They've developed enough because of those daytime temperatures have stayed up, kept them more active, and they can actually start breeding in the fall. They can lay eggs in the fall. Typically, those eggs do not survive to spring. So it's only the spring eggs that are laid that become our problem. We probably need a little bit more research with these type of years. I'll tell you how it would work. If I submitted a research proposal this year, it'd get funded next year, and next year we'd have the worst winter on record, and I wouldn't be able to answer the question for you, is usually how it works. But just be aware, they can develop eggs, lay eggs in the fall. Currently at this point, we do not see that we're seeing double egg laying. Okay? Questions on that? Hopefully, go ahead.
Do you have another picture of an adult? That's a little bit...
I'll have to look. I can throw one up later. I can probably pull a picture up or something.
Are they black?
So they're brown. Yeah, if you come up, they're brown. They're a snout beetle, so they're going to have kind of that proboscis out the front. There's some antennas right here. They're brown, and they have a black stripe down the middle of them. Okay? You'll get into the alfalfa field, and if you're doing any sweeping, there'll be some brown beetles, but they'll have dots on them. Those are not alfalfa weevil adults. Great question. Those are root curculio weevil. So they are a type of weevil. They're not an alfalfa weevil. They're more on a root side. Go ahead.
Last question. I'll find
Yeah.
out a lot more.
That's fine.
The first crop, the grower is just stuck with the weevils, basically.
And that's why we're doing insecticide applications. And so this is specifically for forage. If you get into alfalfa for seed production, they're still battling it as well. And it is an impact for alfalfa seed. So for a grower, you have them. Like if you are raising alfalfa, you have alfalfa weevil to some level. It depends on if that's at an economic threshold or not to warrant if it's worth controlling. And so then we go in and roll in and do an insecticide application to try and control them. We're trying to hit it. If we can time it appropriately, we'd like to hit it when the population is at the highest, in third and fourth instar stages. They're out on the tips of the leaves. They're out on the physical structure of the plant where they're exposed to insecticides. They're vulnerable for that control method. And also at that population. So when we spray, hopefully only once, that it will knock that and suppress that population down so we can get through harvest and save the yield.
Which would in turn not have much leaf retention on the first cutting if this is predominant in the field, right?
If it's allowed to go long enough. if that makes sense. So if we can catch early enough where we haven't had much damage, we will have good leaf retention. If those leaves are skeletonized, it's a drier plant, you'll probably have a lot more shatter and loss as you're harvesting. Does that answer your question?
So we just had a bee pollinator thing in here, So talk to us about how do you kill the larva without killing the bees?
It's the timing of the spray is the biggest thing. So native bees and honeybees have active windows. They tend to prefer bright, sunny days, low wind, which is what we like for spraying too, right? But usually they don't start getting out of the hive, they don't start waking up, even our native bees don't start getting active until about 9:30, 10 o'clock in the morning, and then they're going to bed at dusk. And so maybe like 6, 7 o'clock at night, they're starting to get back into their homes and that. And so in alfalfa seed in particular, how they protect against killing their leafcutter bees that are doing their pollination, they're spraying at night. Most of our forage producers, I don't see them necessarily spraying at night, but they might spray at early morning or late in the evening to try and miss that window. Yeah, good question. Are we good on the life cycle?
I have a question. If you have a cool summer, if it stays cooler, like over the mountain, will they have to go through another life cycle?
If you have a cool summer. No, not necessarily. I should mention,
Summer dormancy?
it'll drag it out.
Stay low?
They'll be active, but they won't develop eggs, if that makes any sense. So a lot of insects, and alfalfa weevil are no different. You can track their life stage and growth and development to temperature. You can plot it on a map basically. They directly correlate. So as that temperature warms up in the spring, you see a lot more activity. It doesn't correlate in terms of like that summertime. So if we have a cool, wet spring, it slows down that development, but we still have the development, right? And into summer, it hits up there a lot of times, and we're talking daytime temperatures, right? But there's very few summers where we hit 70 degrees and stay at 70 degrees. So if you get higher elevations, they don't tend to have as much alfalfa weevil problem, but it's because of the shortness of their year. They still have them, but not to the level of what we see. So Riverton's one of the areas, the Bighorn Basin, and then that southeast part of the state, I see a lot more alfalfa weevil activity because of the higher temperatures. Just they have a longer growing season, right? They're active in the springtime. But yeah, they hit that summertime and they just go calm. They just relax. Doesn't mean that cooler temperature. You'd have to have 2.5 to 3 months of cold temperatures, like 70-degree temperatures, to probably see a difference, to disrupt that summer dormancy. Does that answer your question? Okay. Okay, so I'm a part of a study with UW Extension. It's myself and Scott Schell. Scott Schell is the entomologist on campus for UW Extension. If you ever worked with him, fantastic guy. We're so fortunate to have him. We have collaborated with Montana State University to look at alfalfa weevil and alfalfa weevil management. We were working on another study on alfalfa weevil when this opportunity came up to collect some samples and to look into insecticide resistance. They were getting reports out of Bighorn County, Montana, which is directly north of Sheridan, Wyoming, and having suspected insecticide resistance. That's kind of what kicked this all off. They were able to get a grant and fund this, and so we started looking into it. Just to be clear, for insects, and in all resistance, but in insects, insecticide resistance is a genetic trait. Okay, so it's genetically passed on from one generation to the next. And it happens through. And we're going to talk specifically spraying, but I just want to make sure it's clear that any and all control measures can develop resistance as well. So we're talking chemical today, but you can get into other fashions. And as an example, there was a professor at a university that released a bunch of houseflies into a room, let's say like this, and he'd turn his students in with fly swatters. And he'd give them a limited time in the room for periods of time, and they'd go in and kill as many flies as they could. And what he saw is a population shift of the flies to where they all, majority of them, a high percentage, like 95% of them were on the ceiling. Thus they couldn't be reached with the fly swatters. And so that is a population shift within those flies to that control method. So it happens to all control methods. It's not just insecticide or chemical control, but we want to highlight that today. So how it works is we have a population, all these little guys that are kind of bluish green that have the S on them are a susceptible individual in the population. Susceptible meaning that they are susceptible to the insecticide. Okay? The purple ones are our resistant individuals. We come across with our spray, we have a few left over. Anytime we spray a field, we're not going to get 100% control within that field. There's various variables within that that has that situation. If we have enough resistance, individuals survive those sprays, they breed, the next season we're going to see more resistance within the population. If we keep spraying with the same chemical over and over and over multiple years, that's when we see the resistance in populations come up to levels to where you're actually going to notice it within the field. I just showed 2 seasons in this diagram. It's not that short with weevil anyway, okay? Others, like plants in particular, Palmer amaranth's one of them, 2 or 3 season growing years it can develop resistance. So there are some out there that can develop really quickly. But it's all through genetics. I just want that to be reiterated, because there are things out there on the internet that says that it's because of the chemicals is causing a mutation in the organism causing the resistance, and that's not true. So here's a summary of what that research has found from MSU. What you want to see is the green dots in the western states are susceptible populations that they've been able to identify. Red dots are highly resistant. To understand the difference, they did this category. I just want to highlight this. So susceptible means less than the minimum application rate. So when we have an insecticide, you read that label, they have application rate recommendations, right? These susceptible populations are being controlled at rates lower than that recommended rate on the label, okay? Moderately resistant is between the minimum application rate on the label and up to 10 times the minimum application rate. And then highly resistant is more than 10 times the application rate. That's a lot. That's off-label. You guys aren't allowed to mix at that high of a rate and have control, okay? So the thing I want to highlight on this map is the green dots. We have susceptible populations in every state that was sampled. We also have resistance in every state that was sampled. That's a good and a bad. We have resistance in every state that was sampled. But the positive side of that, the light of that, is susceptible populations still do exist out there. So not everything is resistance, even though we found high resistance. Does that make sense? Questions on that? Okay. They've done a lot of work down here in California, and we'll talk about that here in a little bit. So this is what those larvae look like. As they get into that 3rd and 4th instar stage, they'll actually develop a white line that goes down their back as well. It's real faint in the earlier stages. It becomes more prevalent. How do they determine that resistance level? So they get samples of larvae out of the field. They actually put them in what's called bioassays. So they put them in little test tubes, and they put a certain number of larvae into that test tube, and that test tube's already been treated with that insecticide and dried within there. They're then sealed, left for 24 hours. They come back, put them on a hot plate, and the hot plate makes the larvae move so they know if they're dead or not. So then they count them. What you're going to see in a susceptible population, so this is the percent affected by the insecticide, and this is the dose rate of that vial, right? And so the more susceptible ones, you're going to have a higher kill rate and quicker, right? The smaller the dose size. With the resistant population, it just kind of gets up and flatlines. If you look at this, this is 30 times the label rate. And it's not much different than down here below the label rate. So for you guys, doubling the rate or anything like that is not going to benefit you if you're in a resistance situation. You have a problem, okay? But for you guys, you could care less about a lab. What does it look like in the field? So the field here on the right, this is in Bighorn County, Montana. This side of the field, this is a whole alfalfa field. It was sprayed with Steward, is the chemical. It's an indoxacarb, is the active ingredient in that chemical. So they actually have resistance to pyrethroids. So we'll get into it, class 3A of insecticides. But this was a test strip here that was done with an ATV, did different chemicals throughout that. These areas that are yellow, that's alfalfa. It might look like cheatgrass or weeds or something. That's actually pure alfalfa. That yellowing, they were sprayed, and that yellow is that damage to that skeletonizing of the plants. And it was so severe that they just turned yellow or white, kind of like a straw. If you've ever seen severe alfalfa weevil, you'll know what it looks like. You can see it from the road. It looks like almost like frost damage out there. But the thing is they sprayed these in the same timing as this field got treated, but the resistance went through it. Does that make sense? It's almost like pouring water on them. It had no effect to those larvae within that. The other thing that that's interesting to a nerd like me, I know it's not good for a producer, but you look at you can see the rectangle. That's indoxacarb within that treatment area. It shows how immobile those larvae are. They don't move very much. You can see actual distinct lines there, unlike something like a grasshopper that will crawl and move, right? So once they're in the larval stage, they're there. So if you can get good control on them, they're there. They're going to stay, right? So how do we know? This is the data from that study. This was that control within that plot. Here's that pyrethroid. It's actually higher. They had higher larval populations in those treated areas than the control. Indoxacarb, like I mentioned, Stewart, had good control. Those weevil aren't resistant to Stewart. So the whole population is susceptible to Stewart. And why is that? We haven't been using Stewart. Okay? It's not necessarily a new chemical. It's new to our area. Right? We haven't used it historically. So let's look at insecticides. You guys are probably familiar with modes of action. Specifically, it's becoming big news. Anytime you hear about pesticides or anything along resistance management, modes of action is a communication on pesticides to communicate how that pesticide is controlling that pest. In specific, this is for insecticides. You're seeing it on fungicides, you're seeing it on insecticides, you're seeing it on herbicides. It's all for resistance management. And the reason for that is if you just change the trade name or the chemical active ingredient within that product, you're not always changing the mode of action of how that chemical kills that pest. And so you're not mixing it up. So for example here, we have about 3 different types of insecticides that are commonly used for alfalfa weevil. The first one's up here, clopyrifos. Lorsband is probably what you guys are more familiar with. That's a class 1B. Our pyrethroids are a class 3A. And then this indoxacarb or Stewart is down there on a 22A. So if I move that mode of action, I'm actually changing how that chemical controls the pest. And thus I'm getting rotation in my chemical. If I'm over here with Mustang Max as a pyrethroid as a 3A, and I switch to Warrior 2, those are both class 3A chemicals. They use the same mode of action to control alfalfa weevil. So I'm not rotating my chemical, even though I think I am. Does that make sense? The difficulty. For alfalfa weevil, we have three different modes of action to use on our pests. This is under question. Lorsband, the EPA is looking at banning Lorsband for any food crops. I can't get a clear answer from Wyoming Department of Ag, or Jeff Edwards, who is our pesticide coordinator for the University of Wyoming, or from EPA, if alfalfa for feed, for livestock feed, counts as a food crop. I can't tell if they're trying to differentiate between human consumed food or livestock feed, or if it's all one under EPA. They won't give a clear answer. What the problem with that is this is so far an effective chemical, but we might lose that. Now we're down to 2 modes of action. If we get resistance in an area to a pyrethroid, we're now down to 1 class mode of action. How do we do integrated pest management if we keep taking tools away? And it's not up to, unfortunately, not our choice necessarily. Okay, does that make sense? Any questions on that? Okay, so what we sampled here in Wyoming, these are the viable samples we got in the state for last year, for 2021. This is very difficult. For the research studies that I've been a part of, this is really hard to get a viable sample because first of all, I have to identify a field, get access to that field, get into the field at the proper timing, and then I got to collect between 2,000 to 4,000 larvae. Keep them alive, ship them, and keep them alive in the mail to Montana. They keep them, rear them, and then try and do the treatment. So there's a lot of places there that it goes wrong. Our biggest issue is getting enough larvae sampled from one field. A lot of people say, yeah, I got weevil problem terrible, and I've been out in fields, and it's a lot of work. I've spent hours out there to collect 200 or 300 head of larvae, and that's not enough, right? I'm going to be there all day and never get to 2,000 or 4,000. So we're hitting specific types of fields. Probably most of the fields that we actually want to be sampling or need to be sampling are getting sprayed, right? So then after they've been sprayed, we can't do the study, that thing. So anyway, there was more than this county sampled. These were the 3 that got viable samples last year. This is what we saw. Blue is susceptible populations to a pyrethroid, okay? So pyrethroids in that southeast corner, we're not seeing resistance there. I should have clarified this before. So if you look at this one, it's a permethrin. It's type 1 pyrethroid. There's 2 classes of pyrethroids. So when we're talking about that class 3A as modes of action, there's the old chemistry, type 1, and the newer chemistry, type 2s. Interesting enough, we saw some resistance, moderate resistance to the type 2 pyrethroids. Type 1, and that's because everybody switched to type 2 type pyrethroids. We're using those, we're developing it. So even within class 3A, we're seeing a difference within those chemicals in terms of resistance and still effective. Even though we have some resistance here, we did it over here with a type 1. So that's a benefit for us, but it's not the whole answer, right? The other side of that, trying to get my numbers right. So a type 2 pyrethroid again, but we have high resistance. Different chemical, right? Lambda-cyhalothrin, zeta-cypermethrin. Okay, those are different active ingredients in the chemical, same mode of action, type 2 pyrethroids. And we're seeing a little bit of a difference in resistance there, okay? But what you really want to know, well, that's wonderful, great. So what? What do we do about this? I first want to just make you aware that we have identified insecticide resistance in Wyoming. Be aware of that. Be on the lookout for it. Just because we found a sample or 2 in your county doesn't mean that you have resistance, okay? But what do you do about it as a producer? The best recommendation we have is monitoring. That is the best tool that you have available to you to protect against resistance. Knowing that you have it first is the #1 thing. And it's important in terms of monitoring before and after your insecticide applications. Knowing how much you had before, get your spray on in a timely manner as best you can. Go in and check it afterwards and see if you have, if your chemical had good control. If there's no difference after that, that's when I'd start suspecting alfalfa weevil resistance for an insecticide. Know what chemical you apply so we can start teasing that out. Does that make sense? Many of you might not have a sweep net, right? That's kind of for crop advisors. You can buy one. They're on Gimplers. I want to say they're 30 bucks, something like that. You can use it. You just go through the field. If you don't have a sweep net, you can just go grab stems of alfalfa and take a 5-gallon bucket. Beat them into your bucket and count your larvae that way, okay? Monitoring is your first thing. Our recommendation, and I'm gonna throw the scientific community on the sword for you here. Scientific community is struggling with resistance. Okay, we have information, we understand it to a little bit. We have no clear direction of how to combat it. It is very difficult. You're hearing in the herbicide world, tank mix. Tank mix, tank mix, tank mix, right? Get 2 or 3 different modes of action of chemistry into that tank that's controlling that pest. That's appropriate. Do it. Only for weeds though. In the entomology world, on the insecticide, they are discouraging and not recommending tank mixing for insects. So for alfalfa weevil, stay with one mode of action, rotate that mode of action annually. If you run into a situation where you had a situation where you're suspecting a failure of the chemical, then you want to rotate that chemical. Don't go back in, and if you're going to treat a second time, don't treat it with the same chemical, okay? Unless you can rule out that it wasn't resistance. There's a lot of times I got producers that call me up and say, I got resistance in my field, and I struggle like heck to figure out that it wasn't an application error. Not intentional, but maybe the weather wasn't right, maybe the rig wasn't set well. There's multiple factors that can influence that, but I struggle a lot of times trying to rule that side out is it was an application error, not resistance. Does that make sense? So that's what we have. If you have the ability, I know this isn't always likely, but if you have the ability to harvest early and not take an insecticide application, do that. The negative to that, I have seen a couple situations, not very common in forage hay, where there has been what they call a green bridge to where we've seen weevil pressure jump to the second cutting. Specifically, I saw it in haylage. Somebody decided to take their first crop as haylage. They went in with the haylage machine. They swapped it, put it into windrows. The next day, they ran in there with the haylage machine, chopped it, threw it in the truck, and was off the field spinning the pivot. That field never dried down like a normal hay operation, and that alfalfa weevil that was there, and it was actually a field I sampled, and I couldn't get enough weevil out of it to make a sample for this study. I was like, you're good. There's hardly any weevil in here. You're good to go. They called me in July and said our second cutting was decimated. They had huge circles in their field about as big as this room or bigger that were just dead. And that alfalfa weevil just survived through that harvest mechanism and started foraging on the second and finished their life cycle. So there is those situations. If you harvest early, you can, again, monitor, right? You can see those weevil into the second cutting. It's a possibility.
So in that circumstance, if that's just a one field called a full section, if that individual knows he's got any cases of weevil in there, can he not go in there and do that same scenario and just grind it all or just get it out of there as silage? And will that remove the weevil?
I personally, I have this is anecdotal, right? I have no research to support this, but at least through my observations and what producers are telling me, we have a better ability to kill the weevil through a haying operation than we do haylage. Those larvae are surviving that haylage ability. And I think it's not 100% clear on it, but it's the type of equipment that we have. And it's more on when we go through a swather, we're crushing, right? We're running that windrow, so we're physically killing larvae that go through. And then once we're baling, that compaction of that baler on it, maybe squares or rounds, we're just compressing on that and killing those larvae. They're a soft-bodied insect at that point, and so we're having a lot of kill that way through a hay than I saw, and that was only one instance in a haylage. I just don't think we're physically killing them through the haylage process. You would think so. That thing looks big and gnarly and nasty, but I think the larvae are small enough they just kind of bounce around and lay on the ground till the equipment's gone and get up and get going again, is my speculation. Yeah. But monitor that. But if you have the ability, if you don't get to thresholds where it's worth spraying, if we don't spray, that chemical pressure is not going to be put on that population, and we're not going to see the shift as readily to insecticide resistance as quickly. It's a delay action, right? Again, I don't think the scientific community, all the experts I've talked to across the country, we don't have an answer, right? How do we get this to go backwards, so to speak, okay? Lessons learned from California. This is just a little joke, poke fun at California. Thank God they banned plastic straws. Did it really fix the problem? Right? Just to highlight it, so down there in that southwestern part of California, this is what they are doing. Okay? This is through regulation. This is the type of chemical. So this is that indoxacarb that was class 22. This is clopyrifos. This is Lorsban. That was that, I can't remember the number now. 1B, I think it was. And then these are our pyrethroids, class 3As, okay? This is the number of acres treated in California. This is the year. And what we see, Lorsban got banned through regulation in California. They are no longer allowed to use clopyrifos in California, so that's out of the toolbox, okay? Our pyrethroids are there. They can use them, still being utilized. Indoxacarb, Stewart, is starting to go up. This is sales of chemical, okay? These are two different counties in California. And it lines up exactly with the slide I just showed you, right? Pyrethroids, we saw a drop off in that. Indoxacarb, we see an increase in use. They have resistance to pyrethroids. Indoxacarb, Stewart, is a more expensive chemical than pyrethroids, and they're still switching hard to it. What do you think is going to happen?
New resistance.
We're going to get new resistance. The biggest fear is, what if we got resistance to both chemicals? Now we have no chemicals available at all for control. Don't be like California. Don't just jump on the bandwagon and say, well, Stewart's working, this is what's available, let's use it. Because if we just switch from whatever we were doing before habitually and switch to a new chemical and do that habitually, we're going to end up in the same results. And California's seeing that. And that's what they're expecting to see with this switch. No offense to the farmers or producers there in California. To a degree, their hand's been forced that way, right? And same for all of us, especially with EPA taking one of our tools out of the toolbox. It's tough, okay? There's studies out there like this. This is out of Colorado, where they've taken all these products, screened them. Here's Stewart, Mustang Max, Lorsban, and showed what pounds per acre, what their rate was, and then the control. If you have a susceptible population to any one of those chemicals, you're going to have good control. The minute you start developing resistance to one or more of those chemicals, that's when the control is going to fail. So these studies, the best I can say from the data, they were all conducted in an area that had susceptible populations. That's why we get the result. The chemicals are still effective. It's a matter of what population we're putting that chemical on. Does that make sense? So there's questions of like, how do we do this better? What do we do? There are some people exploring, doing some exploratory stuff. There's other research trying to protect our pollinators, right? They're trying to go after army cutworms. They're doing an early treatment, was what it's called. I can't remember if I don't know if I remember how early they were. Anyway, so they're trying to explore with doing early treatments of insecticide and seeing how much control we have. Can we protect our pollinators? Can we have control of army cutworms and line those up with alfalfa weevil? The preliminary research shows that it's effective. That's good. Here in Wyoming, talk about that in a little bit. Here in Wyoming, we had down in the southeast part of the state, had a producer went out and sprayed. This was about 4 years ago, I want to say. He sprayed the edges of his alfalfa field for herbicide, for grass control. Ended up just throwing in an insecticide in the tank and did a tank mix that way with a herbicide and an insecticide. That year, his whole field was green on the outside border strip that he sprayed. The middle was decimated by alfalfa weevil. So it kind of begged this question, and he was spraying that early in the spring, probably about end of March, beginning April, when the alfalfa was like 3 inches tall, 4 inches tall. That was fantastic news. We then did it again for the next 2 years, and we've had mixed results with that. And it has to do with the timing of the adults, right? So if we can kill those adults before they lay eggs, we don't have a weevil problem. But the difficulty is timing that spray into that field when a majority of those adults are there and prior to them laying eggs. If they've laid enough eggs into that field already and we spray, it was for naught, right? We're still going to have a weevil population. If we get there and spray and the weevil haven't moved into the field yet, that spray was for naught again. We have no way of monitoring how to time that type of spray. There's something there, we can't figure it out quite yet. A lot of people ask me, Jeremiah, why do we have, it seems like alfalfa weevil are worse now than when I remember in the 70s and 80s. The best answer I can give you is since 1975, we have seen nothing but an increase of alfalfa fields in the area. We have more habitat, more food source for those weevil, and not everybody is treating their fields the same. There's people that are not spraying, there's people that are spraying multiple times. It's just a random hodgepodge if you look at it as a landscape. This is the only thing that I can see as a direct correlation to our high alfalfa weevil populations, is the increase in alfalfa fields. Okay? A couple things. So I talked about this one, spraying for cutworms and weeds in the spring. It was effective. We don't know how to time it. There's an individual I've heard of here in Fremont County. You might know him. But they run horses. It sounds like they run dude horses on their operation. They don't do any spraying, and they're plumb happy with their yields. It doesn't mean that they don't have alfalfa weevil, but they never get to a level that decimates his fields or yields. I don't know exactly what he's doing, but everything I can figure out is he's running horses out there, runs them fairly into the spring, March, April-ish time period. That's that time when those adults are moving into the field. And so he is removing biomass, he's removing that litter, he's removing any kind of green growth coming up, and the weevil might be going to the neighbors. And then he gets the horses off, that comes up, and if the weevil aren't in the field and aren't mating, and they're all in the other fields, he doesn't have the pressure. So there's something there, but as you guys know, if you graze those hay fields in the spring, there's something to that, right? You're going to be hurting your yields the farther you go into the spring and grazing on that. And I also kind of wonder if there's something to it with him grazing horses on it instead of cattle. So maybe that solid hoof has a better compaction rate in terms of crushing and killing larvae as they're going through the field. And horses have that nipping ability to graze down and remove biomass completely to the ground and damage crowns too if it gets severe enough. Cattle don't necessarily have that ability. So there's something there too that's all anecdotic. What we're doing at the university, so myself and Scott Shell, like I said, we're going to continue trying to collaborate as much as we can on this insecticide resistance work. It's more of just identifying resistance and where it's at in the state. We're going to try and collect samples again this year. We'll see how well we do. Dr. Rinda Jabor down on campus has finished a couple studies on alfalfa weevil. One in particular is on early harvest. All the recommendations in the literature is like, your two control options are use an insecticide and spray appropriately, or do an early harvest. But nobody says, what does that early harvest mean? Is there a timing to it? How do we figure out that timing? She looked at that. I haven't seen the results yet, but hopefully maybe this spring, like April or May, she'll have time to crunch that data and we'll be able to get that information out here this year sometime. So things that we're working on, that's what I got for you. Don't want to keep you from lunch. I'm here though. Here.
What about diatomaceous earth?
I don't know. It's a good question. I have not seen that. I don't know any information on, I know what you're talking about, so they'll put diatomaceous earth out in gardens and landscapes for certain controls. My two thoughts I have with that is one, where the weevil are up on the plant, it may be hard to keep that diatomaceous earth up there and in contact with them if it had a control effect to it. I don't know if it does. The second thing is here in Wyoming, our winds are terrible, and I don't know how you keep that powder, that dry formulation in the place you need
Could
it.
you slurry it, maybe?
Maybe, but I don't even know if it'll have control on them. I don't know if it'll have the effect. So they say with ants in particular, you can put that diatomaceous earth around their holes, and what they do is they pack that on their body, and it's like razor blades actually. And when they go down into their hole, they get into the brood, and they're actually killing the young and the eggs, is how it does control on ants that I'm aware of. I don't know if that'll have, I'd almost suspect it wouldn't have the same effect on alfalfa weevil larvae, but I don't know. Nobody's ever asked that question, or looked at it that I know of. Great question. I'm here. I'm willing to talk. I'm going to be here through lunch, and then I got to catch a meeting this afternoon, but be glad to chat with you or help out. If you got to go, feel free to run. I think they'd love it if you'd fill out an evaluation.